Welcome back. We're going to start discussing the final stages of aerobic cellular respiration. We're going to go over oxidative phosphorylation. All right, remember with oxidative phosphorylation, we've got two stages. We've got the electron transport chain, and we're going to have chemiosmosis. And I'm going to define on my diagram over here where each of those stages is occurring and what's happening in each of those stages. All right, but we're going to start out with that. And essentially what oxidative phosphorylation does is it's going to capture or tap energy from what are referred to as falling electrons. Because if you remember from our previous two stages of cellular respiration, from glycolysis and the Krebs cycle, we generated molecules. We generated NADH and we generated FADH2, okay? These electrons are at a high level of energy, okay? And we're going to exploit that as we put those high energy electrons into the inner mitochondrial membrane to generate ATP, okay? So what we're going to do when we talk about falling electrons, okay, our electrons are going to go from our high energy state to a low energy state. Okay? That's what it means for the electrons to fall. They're going from high energy to low energy. All right? So what we've got here, I'm going to define where we are here in the mitochondria. Okay? This side of the membrane is going to be our mitochondrial matrix. Okay, so this is the innermost portion of the mitochondria. This is going to be our inner mitochondrial membrane. Okay? And then what we would have, I'm not going to draw the membrane itself. I'm going to put it as kind of a wiggly line up here. This one, this green wiggly line that I did here, this is going to be our outer mitochondrial membrane. Okay? So what we've got is we've got a little bit of space here. This is the inner membranous space, all right? And that's going to become important in the process of oxidative phosphorylation, okay? Because what we're going to do, because we've got some different compartments here, is we are going to generate a hydrogen gradient. So one of the things that's key to oxidative phosphorylation and the electron transport chain is going to be generating a hydrogen gradient. Okay? Because that's going to be key in our ability to generate ATP out of this process. All right? And the way we're going to generate that hydrogen gradient is going to involve our NADH and our FADH2. Okay? So if you notice, I've got kind of some pink blobs drawn in the inner mitochondrial membrane here. These pink blobs are cytochromes. okay? And I have in the PowerPoints the names of each of the cytochromes so that you'll know so that you have that. What I'm concerned about that you know is there are 4 cytochromes here in the inner mitochondrial matrix, and those 4 cytochromes are going to be what constitute our electron transport chain, okay? And I've been abbreviating that ETC. I guess I'll write it out. But this is our electron transport chain. It's where the ETC comes from, okay? So we've got our electron transport chain, and then if you notice at the end here, there's something else, all right? I've got this in your notes as the 5th cytochrome, okay? What this molecule is, this is going to be ATP synthase. This ATP synthase is going to be where chemiosmosis is going to happen in our oxidative phosphorylation. So oxidative phosphorylation collectively, all of the cytochromes associated with the electron transport chain and ATP synthase makes up the process of oxidative phosphorylation. All right, the 4 in pink I've got here, this would be our electron transport chain. The ATP synthase is where chemiosmosis happens. All right, and I'll talk more about what that is here in a minute, okay? But I just want to define where everything is, all right? Now what's going to happen is we're going to now take the electrons that were generated in glycolysis and in the Krebs cycle, and we're going to deposit these electrons into the electron transport chain. So at cytochrome 1, our NADH is going to transfer that electron, and I'll make that as an E here, into our first protein. Excuse me. Our NADH is going to be converted back to NAD. That NAD now can serve in glycolysis and the Krebs cycle to bring more electrons to the electron transport chain. So this is an enzyme, it's renewable, it can be reused, okay? So our NADH is regenerated here. The NADH dumps the electron into the electron transport chain. And then what's going to happen? That electron is going to bounce around in the inner mitochondrial membrane. It's going to kind of jump from protein to protein until it exits through the 4th cytochrome, now as our low energy electron, okay? What's going to happen as that electron bounces through our cell membrane? That's now the energy that goes from being high to low energy is going to allow hydrogen to move across. Oops, let's put this one over here. Our inner mitochondrial membrane. And what that's going to do, I'm going to draw a lot more up here, because this is going to be what generates our hydrogen gradient. So those electrons going into the electron transport chain move hydrogen from an area of low concentration to high. Now when we talked about membrane transport, remember when I said when things went from low to high, you had to put in ATP. In this case, this process, because of the electrons there, the energy in order to move things against the concentration gradient, so we're going from low to high, the energy from the electron is what allows that movement to happen. So what we end up with is a whole lot of hydrogen in our inner mitochondrial membrane space. So between our outer mitochondrial membrane and our inner mitochondrial membrane, we've got lots of hydrogen floating around there, okay? Now, with our electron carriers, I put in NADH here. I said we also have FADH, okay? And that one is a little bit different. It enters at the second cytochrome, and then we go from being FADH2 to FAD, but the same thing happens. We're putting in a high energy electron here. It bounces around. It exits as a low energy electron. It also drives hydrogen to move from the mitochondrial matrix into the space between the two membranes, okay? So that's what we've got going on with our electron, all right? And what's gonna happen, all right? Because I said it's gonna be the electron transport chain and oxidative phosphorylation where we involve oxygen in this process. This is what makes it aerobic, okay? So what we've got, that low energy electron then is going to take our hydrogen, all right? And we'll put 4 hydrogen, so this is balanced, plus our oxygen, okay? And what that's going to generate, going to be some of our water molecules, okay? So when I drew cellular respiration up, remember 2 of the end products out of cellular respiration were carbon dioxide, which we talked about in the previous 2 videos, and water being products that are generated, okay? So this is where the water is generated. That low energy electron is what's going to drive the hydrogen and, let me put the plus sign here, oxygen together to generate water, okay? So this is where water's generated. And that oxygen, where can I write this? I'm running out of space, okay? Oxygen in aerobic cellular respiration is going to be our final electron acceptor. So all of those electrons that we stripped out of glucose are going to end up joining up with oxygen to generate our water in this process, okay? So this is where aerobic respiration, where we need the oxygen, is at the end of the electron transport chain. It pulls, the way I think about it, is it pulls that low energy electron out of the electron transport chain to generate our water, okay? So that's what we've got going on there. So the components of the electron transport chain that we've talked about are what are going on here, okay? The electrons bouncing around in those cytochromes, moving hydrogen against its concentration gradient, all right, to generate a high concentration of hydrogen between our 2 membranes. Electron exits the electron transport chain, and this is where our water is generated. This is where our oxygen is used, okay? So we've got that. Then we've got our last steps here. Let me extend my cell membrane here a little bit, okay? What we're gonna have happen now, okay, those hydrogens are going to want to diffuse back across the inner mitochondrial membrane because they want to go from an area of high concentration to low concentration. And what allows that hydrogen to do that is ATP synthase, okay? I'm going to remove this bar here just so that I can draw in what happens with our ATP synthase, okay? That hydrogen is going to shoot essentially through our ATP synthase. That's going to cause the ATP synthase to turn in our plasma membrane. Our hydrogen comes back across the membrane because that's happening by diffusion. All right, what's going to happen? Our ADP and our phosphate that are hanging out there in the inner mitochondrial matrix are going to take advantage of that energy that that hydrogen is going to generate from diffusing across the membrane. And that's going to generate our ATP. So ATP synthase, okay, ACE, remember, tells you this is an enzyme. This particular enzyme is going to synthesize ATP from ADP and a free phosphate, okay? And with all of the NADH and FADH that we generate in glycolysis and the Krebs cycle. So we end up with 6 NADH from the Krebs cycle, 2 FADH from the Krebs cycle, and then we've got some additional NADH that's supplied from glycolysis and the conversion of pyruvate to acetyl-CoA. All of those that are fed from that glucose molecule are going to be what generate the bulk of our ATP in cellular respiration. So out of that process of chemiosmosis, the hydrogen shooting across ATP synthase to join ADP and the phosphate to ATP generates the bulk of our ATP, okay? So this is where we get our 28 ATP, and essentially the way we do this without investing any ATP is to take advantage of the electrons that are stripped off of the glucose to generate our hydrogen gradient. We're using the power from that hydrogen gradient just like you might use the power from a hydroelectric dam, right? You get the water falling over the dam, that is converted into energy that can be used in your house, all right? You're going from high to low through that ATP synthase, generating the bulk of your ATP. So that's overall how oxidative phosphorylation works. I've got a couple more videos that will talk about what can go wrong in this process of oxidative phosphorylation and how that can interfere with cellular respiration. We'll also have some videos on the process of fermentation, so how we generate energy and ATP anaerobically. Because remember, this process, aerobic, involves oxygen. If you have any questions, please let me know.